Biology 2. Well, it's split up into a variety of different parts. We're not going to have time to cover all of those today in this first hangout. We will be looking at cells, specifically their structures, how they're specialised, the organisational parts, and the processes within. We'll then have a look at plants, plant structure, and photosynthesis. We'll then move on to organism distribution, looking at quadrats and transects. And finally, we'll end on enzymes, catalysts and digestion, how they're used in respiration, and finally, how they're used in industry. We'll look at DNA, genetics, and life on Earth in the next session. So part 1 is all to do with cells, specifically the cell structure we find in both animal and plant cells. And there's a few key things that you need to be aware of for each of them. It's unlikely in a higher paper you'll be asked to label a cell for more than a couple of marks, but they do come up quite often, so please be aware of the following. In an animal cell, which is here on the left, we've got the following key components, the cell organelles. The first one is the nucleus. Now, the nucleus contains our genetic information. It controls the activities of a cell. The second is the cytoplasm. Now, specifically with the cytoplasm, it's where our chemical reactions take place, and enzymes are found within there. Thirdly, we have a cell membrane. We'll come on to membranes later, but essentially it's a process where, or a wall basically, which allows certain things to go in and certain other things not to pass through. It's what we call semi-permeable, and we'll talk about that later. The fourth point is our mitochondria. Now, mitochondria is where our reactions for respiration takes place. It's where the energy is released for every single cell in our body. And finally, we have ribosomes. And with ribosomes, this is where our proteins are made. Of all of these, it's likely you'll be asked about nucleus, mitochondria, and ribosomes, but don't leave out cytoplasm and cell brain. These still could be asked. Now, for a plant cell, they have all the same organelles as our animal. We haven't drawn them in here, but they are still there. So, for example, our nucleus is here. Well, on this particular part of the plant, there are 3 slightly different parts that you just need to be aware of, which I'll draw in green. The very first one is the cell wall. It's what gives a plant cell its rigidity, keeps it well structured. We then have a vacuole. Now, inside this part is essentially where the cell sap, the sugars and salts that the cell is keeping and storing for later, is held. And finally, we have our chloroplasts. Now, the chloroplasts contain a pigment called chlorophyll, and it's vital for photosynthesis, how the plant makes its own food. Now, for plants, again, you could be asked for any of the organelles found within an animal cell. Specifically, though, questions tend to be around the cell wall and the chloroplasts. Now, finally, with all the structures of the cell, you do need to know 2 other specific types. The first are yeast. Now, yeast cells are tiny microorganisms. They have a nucleus, they have a cytoplasm, a cell membrane, but they're actually surrounded by a cell wall. So they are different to animal and plant cells. They've got a combination of a few key structures. Finally, we have bacteria. Now, the important part for bacteria, being the single-celled microorganism that it is that it doesn't actually have a nucleus. All the genetic material is loose. So this is our first part for cells. Please make sure that you are able to label all of them and all the organelles within, and have a very brief description of what each part does. The second key bit around cells then comes into specialization. How the cell is adapted, the processes that it needs to do. And we need to learn a few key ones. So the very first one, which is on the top left, is our palisade cell. Now, these are leaf cells. They're all about photosynthesis. Again, we'll come on to this specifically in a minute. But essentially, we've got lots of chloroplasts. We've also got a large surface area, which means we can pack lots of chloroplasts in there, but also they can absorb carbon dioxide very well. And again, the key components needed for photosynthesis. Alongside, though, this palisade cell, we have another plant-specific specialized cell. And these are our guard cells. Now, guard cells are essentially adapted to open and close specific pores, known as stomata. They work well because they let in carbon dioxide when they're open. Essentially, they change the amount of liquid within them. They become turgid or flaccid, and that will allow carbon dioxide to go in. But there is a downside to it, because if they're open, the plant is susceptible to losing water. And so the plant will have a compromise. They won't be able to be kept open the entire time, which would be great for photosynthesis, apart from the fact water will be lost. So these are our plant specialized cells. There are two others that you need to know for animals. The first are the gametes, the sex cells, the sperm and the egg. Essentially, they're there for fertilization, the sperm finding its way to the egg. And so the specialization here will be a towel for the sperm to swim. And in the egg, it's got a very large cytoplasm. It's got a large energy store. So when fertilization does occur, the eggs can develop into what will eventually become a fetus. Finally, we have our cells, red blood cells. Now, red blood cells are all about carrying oxygen around the body. And to do that, they have a special pigment, a pigment known as hemoglobin. Well, the specialization with a red blood cell is that it doesn't have a nucleus when they're mature. And so that increases the total amount of area for hemoglobin to reside in. And so we can carry more oxygen around the body. Thirdly, you also will need to be able to talk about the basic organization of a cell, a tissue, an organ, and eventually an organ system. So all animals that are large multicellular organisms have organ systems. They have specialized cells that allow different things to happen. And so if we start at the basic level, we have similar cells are organized into what we call tissues. Now, for the exam, classic ones which come up are muscular tissues, tissues which can contract, allowing twitches to happen, movement. The second type are glandular. For glandular, it's the secretion of enzymes and hormones, certain chemicals. Thirdly and finally, epithelial. Now, epithelial cells cover parts of the body, such as inside the gut. So these are the similar cells that work together to form a tissue. And in terms of cell structure, the tissues themselves then work together, different tissues, working together to form an organ. And so an organ is simply different tissues performing a given function. Finally, we can have multiple organs that work together to make an organ system. And that could be all sorts of different systems, such as the digestive system, where we have our stomach working with other parts such as the pancreas, the intestines, the liver. But again, all of those organs themselves are made up of particular tissues, which are themselves made up of similar cells working together. And so this is the cell organisation. Now finally, with the introduction part of cells, you'll also need to know the different processes that take place. Specifically, for the additional science, you'll need to know the word diffusion. Now, diffusion is very simple, actually. It's the net movement, meaning on average, most of these particles move a particular way. So the net movement of particles from a high to a low concentration. In other words, if we look at the example here on the top left, we've got quite a high concentrate of these red particles, a perfume, for example, sprayed in the corner of a room. Well, give that enough time, they will begin to move and diffuse out. And so eventually, they're no longer in one particular corner, but they are spread out evenly throughout the room. So diffusion is the spreading of particles from an area of high concentration to an area of low concentration. Now with that, if we go back to look specifically at our cells, we have one key aspect where diffusion is used a lot. And this is for our cell membranes. Our cell membrane, as we said previously, is semi-permeable. It allows some things to pass through, but other things to not. So in the case here, for example, this might be the wall of a gut, we've got some very large particles, we've got some proteins and some starches. They're not able to pass through our cell membrane until they're broken up, until they're smaller. And so this membrane is semi-permeable. Some things can pass through whilst others can't. And this is very, very useful for things like root hair cells, which are absorbing water and minerals, for gassy exchange in the lungs, or in this example, for the stomach and the small intestine. So that's the content for cells. And we're starting now with a particular exam question taken from some specimen papers across all the different boards. Again, as we go through this, if you're new to how these Hangouts are working, these are recorded. You can watch them at any time. So if you'd like a little bit more time yourself, just pause it and then you can rejoin me. What I'll do as well, you'll see in the top right, there is a countdown timer, and I'll normally let that run a little bit. So here's question 1. We have a diagram of a bacterium, and we need to label A, B, C, and D for the components within the given organelles. And for the second part of that question, the diagram shows 2 cells, a bacteria and a plant cell. Both the bacteria cell and the plant cell contain ribosomes, and we've got a simple factual, what does a ribosome do? So let's have a very first quick look at question 1. Well, this is bacteria. So the first thing we know is there will not be a nucleus. We're ruling that out. Well, A is the inner lining. A is our cell membrane. B is the jelly-like substance within. That's for cytoplasm. For C, well, this is our DNA or our genetic material or our chromosomes. You would have got away with any of these answers, even DNA actually. It's a slightly different form, but it would have counted in this particular GCSE mark. What you wouldn't have got is highlighting that as a nucleus. Finally, for D, D is our cell wall. Now, on this particular question, it's a simple factual point. What are the ribosomes doing? Well, this is where we get protein synthesis. Okay, the second question for this part. Question 2, I'll read it out. I will give you a little bit of time, but we've got to go relatively quick today. So for 1 mark, the plant cell contains mitochondria, but the bacterial cell does not contain mitochondria. Give one other way in which the plant cell is different from the bacteria. And again, it might be good practice in your real exam that you're highlighting as you go through keywords that are going to allow you to compare and contrast the particular question. The second question, both cells are drawn the same length, but the magnification of each cell is different. The real length of the bacteria is 2 micrometers. Calculate the real length x of the plant cell. Give your answer in micrometers. Show clearly how you work out your answer. And finally, most mitochondria are about 3 micrometers in length. The plant cell contains mitochondria, but the bacterial cell does not contain mitochondria. Use your answer to part B, part 1, and the information in the diagram to suggest why. Okay, for our first mark, a plant cell different from a bacteria. Well, we can say that perhaps the plant has a nucleus, or you could say that it's got a vacuole, or you could say chloroplasts, or you could have referred to something about its size. A plant cell is much bigger. Now for our second mark, we need to look closely at the diagram here. The clue is written in the magnification of my plant cell and the magnification of my bacteria. I know that this particular bacteria is 2 micrometers, and they've drawn the plant cell the same length. So to get the answer here, we're going to say that because we've got a magnification of 50,000 and it's 2 micrometers long, we're going to have 2 multiplied by 50,000, but then we're going to divide it by the magnification of the plant. So 2 times 50,000 is 100,000, divided by 500, the answer is 200 micrometers. Finally, with that information, the plant cell contains mitochondria, but the bacterial cell does not. Why? Well, if we have a look, our bacteria cell is simply too small. The mitochondria are essentially the same size as the bacteria. They wouldn't fit. And that's where we're getting our final mark for that particular question on cells. Okay, the second part is all about plants. We're looking at plant structure. And I'm just having a quick look on Frog, and it doesn't appear that anyone's got any specific questions so far for the cells. So hopefully that's relatively factual and relatively easy as a starting point. Well, for the first part of cells, we're going to need to know, first part of plants, we're going to need to know the structures. But before we get there, I guess the most important thing that we need to know for the additional science paper is what are plants doing? Well, essentially, they are photosynthesizing. They are producing or releasing their own energy. And they're doing that by getting carbon dioxide, water, some light, and they're converting that to glucose and some oxygen. And if we're going to balance, like so. Now we'll talk about all of that photosynthesis in a minute or two. But this is what we're looking at. Plants having an ability to photosynthesize. And with that in mind, their structures will hopefully become relatively clear. So the very first part we need to look at for our plant structure is the following. We find that actually we have a very good ability for the plant to photosynthesize. It has what we call a mesophyll layer. Specifically, the mesophyll layer has got lots of chloroplasts. Lots of these palisade cells from earlier. They're able to photosynthesize. They're able to get in light with their chlorophyll. The second part from that, we have two vessels, the xylem and the phloem. They're there for transport. The xylem is dead material transporting water, and the phloem is live material which is able to transport sugars through the plant. So we've got a mesophyll layer for photosynthesis. We've got xylem and phloem, which are transport organs. And finally, we have an epidermis. Well, the epidermis is essentially a covering. It's allowing for some light protection for the leaf. There are also other key parts. If you notice from earlier, we have ourselves some pores, some stomata, and they're surrounded by guard cells that are going to allow carbon dioxide in. But unfortunately, as a byproduct of that, they will allow water out. So the basic structure of a plant is there to allow photosynthesis to occur. Lots of chloroplast-rich cells at the top, a waxy cuticle, an epidermal cell, some kind of covering for protection, and finally some vessels and some guard cells, some stomata. Now, one of the classic questions you'll be given for anything to do with photosynthesis, along with the equation for photosynthesis, will be what limits it, what increases it, what makes it go quicker, what slows it, what stops it. And so this is our equation down here. We're taking our carbon dioxide, our water, with some sunlight falling upon chlorophyll, and it's going to produce glucose and oxygen. The classic graphs that you're going to need to draw essentially are something like this. You're going to have yourself a rate, so I'll draw it in a slightly different pen so you can see it. You're going to have yourself a rate of photosynthesis, how quickly it's going, or how much is being produced. And then you're going to have some difference. So this could be the amount of light, or it could be the amount of carbon dioxide, or perhaps the amount of water, or even the temperature. And you're going to need to graph for each particular one what's going on. So let's have a very quick look. Our first one here on the left-hand side is light. We'll notice that as we're increasing the amount of light, we're increasing the rate of photosynthesis. But we get to a point where that no longer occurs. It doesn't matter how much more light you give that plant, it will not photosynthesize anymore. And in this case, something else is limiting it. There is another factor needed for photosynthesis that is not allowing that plant to grow as quickly as it might. Chances are it's one of the other reactants needed. Perhaps carbon dioxide, perhaps water, perhaps even the temperature. So our first graph is very simple, light and photosynthesis rate. Now carbon dioxide is exactly the same graph. As we increase the level of carbon dioxide, we will increase the rate of photosynthesis. But again, we'll get to a point. And at this point, increasing carbon dioxide makes no difference to our rate of photosynthesis. Something else is limiting it. Perhaps this time, the sunlight is limiting it. Finally, a slightly different graph, but our temperature graph for rate of photosynthesis. Now here, if you look, we're increasing steadily our rate of photosynthesis like we did in our previous graphs. But what we find here is rather than it tailing off, it actually drops down. And this is because at too high a temperature, the enzymes will denature. They will no longer work. And so the plant will not be able to photosynthesize. So with that in mind, and as before, let's take a look at a couple of questions. And again, you might want to pause this and take your time answering them and come back later on. Well, question 1 is a factual question. It's an equation for photosynthesis. Question 2, some students investigated the effect of temperature on the rate of photosynthesis. In pondweed, they set up the apparatus and altered the temperature using ice and hot water. They count the number of bubbles given off at different temperatures. The question is, why do the students use a water bath? And then we've got a particular graph which we'll look at. Well, very simply, for part 1, this is our photosynthesis equation. So we've got water with our carbon dioxide and our light energy, and our byproduct is oxygen. For this particular question, when they're saying that they are using a water bath, they're doing that because a water bath will keep our temperature constant. They are looking at different temperatures, but you always want that temperature to be constant. For the second question, we're going to look at the graph. And again, I've just placed it over here. It'll be clearer in your exam. It should follow or should be just above each particular question. Explain the shape of the graph between 22 and 27 degrees. And this is 2 marks. And then the second part to this, a farmer wants to grow lettuces as quickly and cheaply as possible in winter. At what temperature should he keep his greenhouse to grow the lettuces as quickly and cheaply as possible? And then an explanation. Well, if we take this graph to begin with, we find that as the temperature increases, the number of gas bubbles increases. And that's a sign those gas bubbles will be oxygen. So that gives us an indication of photosynthesis. But they're specifically saying between 22, which is around about here, and 27. Well, the shape at this point is that it isn't actually changing. So our graph initially increased, but at this point, something is limiting the ability for photosynthesis to occur. So it's not increasing. Now, that's something else, could be a variety of different things. Could be carbon dioxide. It might be the water, potentially. You're getting your second mark from one of the other limiting factors. First of all, by saying the graph is not doing anything, it's not increasing in its photosynthesis, or the number of gas bubbles aren't increasing over that given time, and because something else is limiting it. At what temperature should the farmer keep his greenhouse for the benefit of the lettuces? Well, ideally, we want that at 22 degrees. The reason for 2 marks is this is where photosynthesis is at its maximum. But anything above this temperature, you're wasting your money. You're wasting heat. It's not making the plant grow any quicker. And so you're simply wasting money. So that's plants. I have just had a couple of questions come through. One of the questions is regarding limiting factors. Would CO2 also be a limiting factor as light begins to increase too much? Absolutely right. Our limiting factors would be for a light graph. So as we're increasing the amount of light and we're looking at our rate of photosynthesis, we would find a graph like this. And at this point, Something other than light limits it. A classic answer there would absolutely be carbon dioxide. You might also say water, and you could get away with saying temperature, although the graph doesn't really show a temperature limiting factor as it would dip down. I would stick with a classic example of carbon dioxide and light. So as a rule of thumb, if the graph is to do with light, limiting factors, carbon dioxide, and then vice versa. Okay, everybody, we're now going to move on to the third part, which is organism distribution. And on the face, it's actually quite an easy concept, but they can make it quite a tricky exam question depending on how they're asking. So essentially, organism distribution is simply saying who lives where do we find things, how are they spread about? And to get that with some kind of data, rather than just saying, I think there are some things over here, to actually quantify that, we use quadrats and transects. Now, in terms of the questions, they're probably going to ask you to actually explain how you would go about setting out a quadrat and how you would go about setting out a transect. Well, very roughly, if you took something like this on the screen, if I had myself, for example, a field, in this case it's a yellow field, and I had a certain number of flowers distributed throughout. And the question might be, how many flowers have I got? Well, it actually becomes a very difficult thing to answer if my field is large. In fact, even if my field was 10 meters by 10 meters, my total area is 100 square meters. And so that might take me quite a long time to go about and count them all. So the process is to find out what actually affects this organism distribution. How many are there? What factors are affecting it, be them biotic factors, are there certain animals, there's lots of competition, or perhaps there's abiotic factors. So we might have different temperatures, we might have different soil conditions. Well, this would be how we would go about using a quadrat. A quadrat is essentially a 1 by 1 square. So it has an area of a square meter. And essentially what we do is we place our quadrat in given areas. However, it's quite important that we don't just deliberately place them in specific areas ourselves. That wouldn't be fair. And so what we do is we take our total area of plot and we try and divide it up. So we might say in this particular area, we're going to divide it up, on paper at least, into 100 square meters. Now, I haven't quite drawn this perfectly, but hopefully you get the gist. So this area is area 1, 2, 3, 4, 5, 6, 7, etc., 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 all the way down to what would eventually be 100. Well, for this to work scientifically, we would then have a way to randomly generate some numbers. The more numbers we do, the more times we place our quadrat down, the more valid our actual data set will be. So for example, I might have a random number generator that generates the number 1, 7, 24, and 99. I'd place my first quadrat in area number 1, my second in area number 7, my third in area 24, wherever that happens to be, and finally in 99. All I simply do then is count the number of flowers in my area. So I might have 1 here, 2 here, none in here, none in here. So I had 2 in 4 different areas. So I would say, on average, of those 2, I've got myself 2 in every 4 sampled, which means, roughly speaking, I've got about 1 in every 2 quadrats I place down. And so all we would do at that point would be to multiply up. If for every 2 quadrats I have 1 flower, and I've got myself 100 quadrats in total in this area, I would guesstimate that I've got 50 of these given flowers. Now, clearly, the more times you place that quadrat down and the more times you work out the average for where you've placed it, the more accurate you're going to be. If you simply do 1 quadrat and you place it in 1 area and you didn't find any, you would be led to believe that there would be 0 flowers. And likewise, if you did 1 quadrat that had 1 flower in it, you'd be likely to theorize that there'd be 100 flowers. So the more you do it, the more accurate you will become. But the rule is, you find out your total area of plot, you then place your 1 by 1 quadrat down randomly and multiple times, and then you simply work out the population size by finding the mean number of organisms in your quadrats and how many times those quadrats would fit in the area. Now, with a transect, it's a similar principle. A transect is essentially a line that runs through a given area. It allows us to study what organisms are distributed through a particular place. And so we would get ourselves a tape measure, would run out our line, and we would count at given areas the number of flowers within. So if I had myself my plot that looked like so, I might draw my transect line running down, and every 2 meters I'd place my quadrat and I would count what things are in. Now, it's important that actually when you do a transect, it will flow wherever the sampling area needs to go. So, for example, if we had ourselves a grassy bank, but that also went into a river, our transect would run along the line, and we would still do our 2 meters, because this will identify the difference in our distribution of organisms. If I was looking at snails, for example, I would probably find my terribly drawn snails are on the banks, but they wouldn't be in this area. And that's probably because I've got myself a river. And this also includes if we have ourselves something like a big tree. Our transect line will continue going up into that tree. And so we might find that actually in this particular part here, we get a much greater amount of insect life because we have a completely different distribution based on the habitat. So transects and quadrats are all about collecting organisms and collecting data for them. So let's have a look at question 5. Question 5, some students investigate the distribution of some plants growing in and around a shallow stream. They sampled along a transect line, and the diagram shows their results. The first question, name one species that grew only in the driest conditions. Well, if this is a river, our driest conditions are going to be on the edges. And so looking at all the different organisms, we would find that our white clover is the only one that is found in the dry conditions. So this would be white clover. Our second question, only one species grew in the marsh, the swamp, and the aquatic zone. What species is that? Well, we're looking at an organism that goes in all three areas. And the only one we find here will actually be the red sweet grass. It's in all three different zones. The common duckweed isn't. It's on both sides of the swamp, but they're both swamp. So this is our reed sweet grass. Oh, I can't spell. Finally, duckweed grows floating in water. What evidence is there for this in the students' results? Well, if we look at the duckweed on the diagram, we actually find that the duckweed here is in the swamp, it's in the aquatic zone, and it's in the swamp. But it's never found in the marsh. So how do we know that it grows in floating water? We don't find any in the marshland. It isn't present. Or conversely, we only find it in the swamp or the aquatic zone. Okay, question 6. Now this is a classic 6-marker answer that we find in the additional paper. And again, you'll be expected for using your good English, your spelling, your punctuation, your grammar. They're asking how you would use a 1/2 meter by 1/2 meter quadrat and a 30-meter tape measure to obtain similar data to that shown in the diagram from a previous one, looking at perhaps marshland, aquatic zones. Well, you're going to get a couple of marks here for your spelling, your punctuation and grammar. In fact, you're going to be bounded into 1 or 2 marks, 3 or 4, and then 5 or 6, depending on your spelling, your punctuation and your grammar. But regardless of your spelling, punctuation and grammar, you're still going to need to have 4 or 5 good key points to be aiming for the large marks. So the marks will essentially be as follows. We're doing quadrats with transects. Well, 1 mark will be to say, tape out your transect. Our second mark will be for our placement of our quadrats. It might be every 2 meters, for example. You also need to ensure that you are saying that you're going to place your transect across the stream. We don't stop simply because a stream is there. You then need to be talking about counting the species within each quadrat and tallying them up and adding them together. And based upon our interval part, we'd repeat that along the stream to allow finally for some type of comparison. So essentially, we're placing our tape out, we're placing quadrats at given intervals, we're counting what's in each one, and based upon that information, we'll have some kind of understanding about what lives where and where our rivers might be or other environmental factors. Okay, the final part for today's session is enzymes. Now, enzymes are one of those classic questions that come up every single year, and they can be asked in a variety of different ways. But essentially, we're going to be breaking it down into 2 parts. What is an enzyme? How does it work in the human body? And then how does industry use enzymes? So the very first part to know is the following. Enzymes are catalysts. They're biological catalysts. They speed up reactions. Now, specifically, like catalysts in chemistry, they don't get used up. So they increase the rate of reaction, but they're not used. They don't change. And they have special shapes, and this diagram here shows it well. Essentially, they allow the active site of different substances to normally be brought closer together. The enzyme allows the substance to break down, and it works much more quickly than it would if it were to be left on its own. So they speed up our reactions, but they are relatively delicate. They can be massively affected by both the pH that they're found within and the temperature. We use this word denature. And so with our pH, there's always an optimum pH for a particular enzyme. And that's the same with temperature. There's an optimum temperature. So humans are 37 degrees, thereabouts. Our enzymes are working best around this temperature. And for both, if the pH becomes too strong or too weak, or the temperature becomes too great, our enzymes might find themselves denaturing and not working as effectively, or even completely breaking. Now, specifically for that, different enzymes work at different temperatures and different pHs. So enzymes found within the stomach, the small intestine, will have a tolerance to a lower pH than enzymes found within our mouth. And conversely, some organisms, for example humans, that function at 37, their enzymes will work very effectively at that. But any hotter, they'll begin to denature. But there are bacteria out there that can comfortably survive at 200 degrees centigrade. And so their enzymes are comfortable at that particular point. Now, finally, with enzymes, it is important that you know 3 key parts to do with the human body. And these are amylase. Amylase breaks starch to sugar. We find it in our salivary glands, pancreas, the intestine. We then have protease, which breaks down proteins to amino acids. And these are found in the stomach and the pancreas and the small intestine again. And finally, we have lipase. You'll notice they've all ended in A's. And lipase breaks our lipids, which are our fats, into fatty acids and glycerol. And these are found in the small intestine and the pancreas. Finally, with all of that, there is one other chemical that you need to be aware of when we're talking about the human digestive system, and that's bile. Bile is produced in the liver and stored in the gallbladder, and it works in its main place in the small intestine. And essentially, it acts as an emulsifier, a little bit like fairy liquid. It breaks the fat into tiny droplets. It does actually also neutralize the pH a little bit as well. But essentially, it's breaking these lipids down into smaller, more easily processed parts. Now, the second part for enzymes is how they're involved in respiration. A bit of a tenuous link here. The point being, you need to know 2 parts. First of all, respiration. Well, respiration happens in almost every single cell in the body, certainly every live cell. And it's the releasing of energy. And it comes in 2 guises. The first is our classic aerobic. Aerobic is respiration with oxygen. So we get our glucose, we have some oxygen combined, and we produce carbon dioxide and some water, and we release some energy. And we get lots of energy, and the only issue here is we're building up carbon dioxide. But that's not a huge issue. We can deal with that very easily. The second type of respiration is anaerobic. Now, anaerobic is without oxygen. So when you dive into a swimming pool and you're swimming underwater, or you hold your breath, you don't instantly fall over and it's game over for you. You can still function. Now, we don't do this normally because anaerobic doesn't give us anywhere near as much energy. And the other problem of anaerobic is we take our glucose, but without the oxygen, we can't make carbon dioxide and water. Instead, we get our small amount of energy, but we also get lactic acid. Lactic acid gives us cramp. It causes us to have an oxygen debt to try and remove it. So that's the enzymes within the human body. The final part is enzymes in industry. Now, they're highly used and very effective. So we find them in biological washing powders. We find them in less calorific drinks. Drinks that are very sweet, but they don't seem to have a huge amount of sugar in. Enzymes such as isomerase, which takes glucose and breaks it into the much sweeter fructose. It's much sweeter, so you don't need as much of it. And so you can absolutely reduce the amount of sugar in that drink and still keep the sweetness. The biological detergents that work brilliantly at 30 degrees centigrade. So your washing machine can be on a lower temperature. If it's on a lower temperature, it's cheaper. And finally, in baby foods, we find enzymes that are breaking down large proteins into the amino acids so the babies can digest them easily. Now, with those enzymes, there's many advantages. Mainly, they're speeding up our reaction. Our reaction takes less time, so our costs go down. They also work for a very long time. The downside is some people are allergic to particular washing powders. The biological washing powders will make people itchy. The enzymes are also easily denatured. So you need to be careful on your conditions within industry. Finally, the actual enzymes themselves are often quite expensive to produce, and they're also quite difficult to keep. If you're breaking down glucose into fructose with an enzyme, you then need to separate that enzyme before you sell your fructose drink. And that can be quite an expensive session to do. So, these are enzymes. Here's our final question for our enzymes topic. Part 1. The diagram shows the apparatus used to investigate the digestion of milk fat by an enzyme. The reaction mixture contained milk and the enzyme. In experiment 1, bile is added. In experiment 2, an equal volume of water replaces the bile. We don't have bile in it. In each experiment, the pH is recorded every 2 minutes. We've then got our diagram here. We've got experiment 1 with bile, and we've got experiment 2 without our bile, and we're looking at the pH over time. The question, milk fat is a type of lipid. Explain why the pH changed in experiment 2. Well, the milk fat is a lipid. There's our clue. The lipid is going to be broken down by the enzyme lipase. And in doing so, it's going to make glycerol, if I can spell, but it's also going to make a fatty acid. Because we're making a fatty acid, the pH is going to become more acidic. It's going to go down, and it does. Finally, the last question of the session. And again, I've repeated the results table here on the left-hand side for ease of use. In experiment 1, calculate the average rate of fall in pH per minute between 4 and 8 minutes. Show clearly how you worked out your final answer. Well, we start with a pH of 8.7 at 4 minutes, and by 8 minutes, it becomes 7.7. That is a fall of 1. But it's taken us 4 minutes to get there. So per minute, we are going down by a pH of 0.25, or you would have got away with saying a quarter. And the last question. The average rate of fall in pH per minute for experiment 2 was 0.1 units of pH per minute. Explain the difference in rate of fall in pH when bile is present. Well, bile is giving us an optimal pH for our enzymes to work. And so because of this optimal pH, the rate of reaction is big. And because of that, the lipid is getting broken into fatty acids quickly, and we'll find that the pH will drop quickly. And that's what it's doing. In experiment 1, our pH drops quickly in comparison to when we don't have the bile, because we've got an optimal pH. Lipase can work well. Ladies and gentlemen, that's the end of the session for additional biology. But it's the first part. We will go for part 2 later, and we'll give you the time shortly. But again, don't forget, this forms just a small part of your revision sessions. Included should be your revision cards, the revision videos, such as using those found on Revision Monkey. There's the Brainscape cards, the CGP books, looking on Nodes with Science for past papers, and classic techniques, speaking to people, asking one another questions. I hope you found it useful. I will be online for the next half an hour or so. So if there are any more questions, please let me know. Have a lovely evening.